The root of the word homology means same or similar. And so homologous structures are structures that are similar in related organisms because they inherited that trait or structure from a shared ancestor. This is the opposite of analogous structures. Analogous structures occur due to a similar selective pressure rather than due to shared ancestors passing on the trait. So homology helps us understand change over time because organisms may have similar structures even if they have changed in slight ways. So for example, if we consider for the 5 digits of our hands, we will see this homologous 5 digits in many, many different organisms, even when those 5 digits have changed in how they are used. So traits inherited from shared ancestors can be anatomical, like this example I've given of the 5 digits. It can also be developmental when we look at what an organism looks like as it's forming. And it can also be invisible to us, but still present in the DNA or the protein sequence of an organism. So bone structure is an area where we see quite a bit of homology in modern vertebrates in particular. And so the essential structure is homologous in that you see very similar basic format of bone. So taking a look here at the human arm, the turtle arm, the bat wing, and the whale flipper. And so you see the same basic bone layout. One large bone, 2 smaller bones, and then the small bones that make up the digits. And that same bone pattern is repeated in all of these different organisms, and you can see that highlighted in color. And that's even when the use of the limb has radically altered. So in humans, our arm isn't used for weight bearing. In a turtle, the arm is used for weight bearing because they walk on all fours. A bat uses this arm for flying, and a whale uses this arm for swimming. So all those different uses suggest that this particular bone structure is not needed for the use, but is rather inherited and passed on from an ancestor that had this bone structure. So the ancestor had this basic bone layout, and it was modified as it was passed on to different organisms. And so that's homology. They inherited this structure from a shared ancestor. So sometimes a structure that we inherit, a homologous structure that we inherit, can actually have no function, not even a modified function, but no function anymore. And these are called vestigial structures. So vestigial structures are anatomical homologies that we inherited from an ancestor, but that have no apparent function anymore. So this can include things like, for example, our tailbone. We don't have a tail anymore, so the tailbone is just a remnant. Some people consider wisdom teeth a vestigial structure. They're really not necessary for their use anymore, but we still have them. Things like the appendix used to be considered a vestigial structure, though they're now starting to think that the appendix may be important for our immune system. So it may not actually be a true vestigial structure. So anatomical homologies can be useful for comparing organisms and helping understand their evolutionary pathway, and they also show us change over time. So this bone structure, basic bone structure, changing in all these different mammals. Developmental homologies are similarities in organisms during their developmental stages, so like when the embryo is forming. And so that they can show similarities between organisms that don't seem very similar once they're adults. So in this example, we see a chicken embryo and a human embryo. And so a chicken and a chicken adult and a human adult don't have very many similarities in their physical forms. I think you could safely say that. But when you look at the chicken embryo and the human embryo, you see some similarities. So, for example, the tail and these pharyngeal slits, which and these traits we'll talk about later on, very later on in the story of evolution. But these are present in both of the embryos, and they're present there because of shared ancestry. So the chicken and the human share an ancestor way back in time, and that ancestor had a tail and pharyngeal slits, and so both of the embryos have those today, even though we don't have those in the adult. And so they show that change over time. We shared that trait, and we don't share it as adults anymore. And the third type of homology is molecular homology. Molecular homology is similarities at the molecular level, so that includes molecules, so not physical structures, but those micromolecules in the body. So things like proteins, DNA, it could also be other structures like fats and carbohydrates, or even biochemical pathways. So molecular events that happen in the cell, like cellular respiration, for example, or photosynthesis is another example. So some examples, well, if you look at living things, you see as one level of homology is that all living things use DNA to store information. And so this trait connects every living thing. And in fact, we consider it one of the defining features of life because it is so homologous. So what that suggests is that the very first cells, the very first ancestors of everything used DNA to store information, and all living things that came after that inherited that trait. Glycolysis is also shared among a lot of living things. So it's present in bacteria and in humans and all kinds of other organisms, virtually identical. So again, this suggests that the earliest cells had glycolysis and they passed it on to all later organisms as a homologous trait. One of the interesting things about molecular homology is that you can actually quantify it, right? So this word quantify, oops. And what that means is to give it a numeric value. And we like numeric values because we can compare them easily. So when I think back on the arm example, right? And I say, okay, the human arm is like the bat arm, is like the turtle arm, but which one is more similar? Is the human arm more like the bat arm or more like the turtle arm? Well, when you look at those pictures, you could try and use the picture to decide that, but each individual might decide that differently because it's very subjective. It's not a number value. It's like, I think this one looks more like that one. But if I can turn that comparison into a number comparison, like the human has this number and the turtle has this number, now I can start to make a more easy comparison that's less dependent on who's looking at it. And so when we look at DNA and protein sequences, in particular, we can turn that into a quantified value. So for example, if I want to compare the human to the rhesus monkey, I can look at a protein sequence. So these are amino acids. So this is a protein sequence. Here's the human protein sequence. Here's the rhesus monkey sequence for that same protein. And there's just one difference, okay, in the section that's highlighted here. So when we look at the whole section of the protein, there's actually a 95% similarity to the human protein. So we call this 95% homologous. So the rhesus monkey's protein sequence is 95% homologous or same as the human. So comparing it to human. The green monkey is the same, so also 95%. But if I go down here and look at the chicken, it's only 53% homologous. So now it's very easy for me to say, well, the monkey is 95% homologous to the human. The chicken is only 53% homologous, so the monkey has more similarity to the chicken. There it is in big hard numbers that anyone, no matter who was looking at those numbers, would interpret the same way. So the percent homology is useful to us because it relates to relatedness. So the higher the percent homology, the more closely related two organisms possibly are, as a reasonable hypothesis, okay? So if you're only looking at one protein, you can't conclude entirely, but it suggests that, okay? So from this data, I would be able to hypothesize that the monkeys are more closely related to the human. The rabbit is second most closely related. The dog is third most closely related. The chicken is next most closely related, and going all the way down to the Congo pufferfish, which is the least closely related to humans, based on this percent homology of this particular protein. And so that's the very useful thing about molecular homology. So to wrap up this concept of homology, let's look at an example. What I have pictured here is the sugar glider and the flying squirrel. And you can see that, and this is where the name sugar glider comes from, or flying squirrel, is that they have this tissue here that lets them glide between trees. So they have limbs for regular movement. They don't fly, so they don't flap that, but they can jump and glide from tree to tree. So it helps them jump and travel further than they would be able to if they could just jump. The sugar glider and the flying squirrel are from different continents. And so these continents separated from each other before either organism existed. Okay, so are they close relatives? And is the gliding trait homologous or analogous? So first of all, are they close relatives? Well, as it states here, the continents separated from each other before either organism existed. So that suggests that they are not close relatives based on this similar trait. So they are not close relatives, even though they have the gliding trait. So that means that the gliding trait did not come from their ancestor. They must have gotten it due to a selective pressure, right? So the gliding trait is not inherited from a shared ancestor. If it's not inherited from a shared ancestor, then it is not homologous, because the definition of homology is similarity due to shared ancestry, okay? Instead, it's an analogous trait, which is the result of convergent evolution. So they got this trait individually, independently, through two separate paths of evolution because of a selective pressure, the benefit of being able to move from tree to tree faster. And so here's a representation of their evolution. So they both come from a rat-like common ancestor. That rat-like common ancestor colonized the Australian region, and then it also was present in the other continent, right? So there's the big continent, there's Australia, okay? And then the placental mammals would go on to create a line of rodentia, so this includes rats, etc. And then some of those rats would become the flying squirrel, or would give rise to the flying squirrel. Over in Australia, we had the marsupial mammals. Remember, that means a pouch, and that would include things like kangaroos and koalas and things like that. And from that lineage, there would be the sugar glider. And so they got their trait, that gliding trait, through two distinct lineages that are not close relatives. And so they are analogous structures that are the result of convergent evolution.